JIS IASR. So it's just a newborn institute which haven't celebrated its first birthday yet. And it's a dream project of Padma Shri Professor Ajay Kumar Rai, who had a vision to create a research-based postgraduate institute for encouraging translation-based research and to offer job-oriented courses for students. So what is translational based research? That means the research which is not for publishing papers only, which is for the use of mankind, our society. So it's kind of whatever we are working on, it should be useful, because we are a country from like India. We spend so much money on research, so it should be useful for people, not only in India, but all over the world. So that's the vision. And also he wanted to create some institute which will make a mark nationwide. So that's his dream, and he seeded that dream into us. So JIS IASR has 3 centers: Center for Data Sciences, Center for Health Science and Technology, in short, CHEST, and Center for Interdisciplinary Sciences. And as I already mentioned, I am here representing Center for Health Science and Technology. So regarding CHEST, we can say that we are unique in Eastern Zone, not only in Kolkata, in the entire Eastern Zone, and the facilities we have is rare in India. So our laboratories, that is the foremost thing to mention. We have a state-of-art laboratory with all next generation and modern equipments, including the Illumina MiSeq next generation sequencer, which is a must one for these days research in genomics. And our center have, not only our center, the whole JIS IASR have internationally experienced faculty team. So Professor Ray contacted us, and he picked the best from all over the world. So the faculties here are have got their training throughout the world. So the faculties are from USA, from different countries of Europe, and Korea, Australia. So it covered at the whole world. And so we have seen all of us, all faculties here in JIS IASR, we have seen the best in our field, and we are committed to bring the best here in JIS IASR. And the programs we are offering are very much need of the hour. That is, the CHEST is offering the bioinformatics program. And also we have a combination program where medical microbiology and bioinformatics are held together. So these two subjects are so interwoven, and experience in both of these subjects is very much required. So today's world, where health and medicine have improved so much, and the advancement of the technology has generated so much data, and I am sorry to say, but there are less efficient people to tackle that kind of data and take out meaningful information from that. So it is really a need of the hour, need a field to pursue, where you can have efficient bioinformatics person with an exposure of medical biotechnology, who can handle this huge amount of health and medicine data very well, and take out meaningful information, which will be the very much useful for our society. And here in our center, there are many of our faculties having prestigious high budget research grants. And they are committed to pursue quality level research. And it last but not the least, most of them have published peer reviewed top tier journals. In short, I can say that we are unique and well equipped in our field in Eastern Zone. So talking about the research, here are some topics that we are dealing with right now. So like bacterial virulence and antibiotic resistance, big data of pathogen evolution in nature, image processing and computer vision, post-pathogen interaction, human microbiome and metagenomics, medical microbiology, medical imaging, and so on. So these are the few we can name, and we have several ongoing research projects funded by DBT ICMR, DBT Wellcome Trust, DBT Ramalingo Swami Fellowship. So these are the few that we can name. And as I mentioned before, we have all advanced level equipments in our biotechnology and bioinformatics laboratory. So everything that today's modern level genomics lab should have. So we have everything, and along with the wet lab, we have the high performance clusters because our center have very efficient bioinformatician. So it's a good combination of wet and dry lab, and we have everything, every instrument, every facility we need to pursue high quality research, and it will be a great exposure for the students. So our molecular genomics core facility offer all kind of DNA sequencing, since we have the modern equipment. And so we could do small genome sequencing, chip sequencing, targeted gene sequencing, 16S metagenomic sequencing, and so on. There are so much possibilities to explore, and that because it's a very less time to talk about. So I would not go into very much detail. And these are the faculties in Center for Health Sciences. And the courses we offer are 2-year MSc in Medical Biotechnology and Bioinformatics. And along with that, we have 2-year MTech in Bioinformatics. And of course, we have PhD program in related biological field to some of the areas to name, like bioinformatics, medical microbiology, cancer biology, biomedical engineering, etc. So this is about what we have here in JIS IASR Kolkata, but one cannot clap with single hands. So we need students. We need good, enthusiastic students who are interested to pursue quality level academic programs and are looking for job-oriented course. So so that's why I'm mentioning here, our admissions are open, and together only we could make the dream of Professor Roy and all of us come true, and one day make the JIS ISR to reach the top, and along with its students. So together we will target the top. I can talk on and on about our institute, but I think I should stop and get into the actual topic of today, which is bacterial signaling and infectious diseases. So I should mention here, my talk is composed at a very basic level, basically designed for the students at BSc level. And as our program, when we design our program, we had that vision in mind that anyone coming from any field should get comfortably incorporated here. So that is also the vision in in planning my talk here. So I'll be dealing with very basics of microbiology and bacteriology. So if anyone feels bored, please bear with me. So talking about bacteria, what the picture comes in our mind? Oh, some notorious organism making trouble for us, creating, which can only be seen under the microscope, right? That's the only thing we can think about bacteria. So bacteria are, I beg to differ that bacteria are beyond that. So just a few preliminary information about bacteria. Bacteria are prokaryotic single cell microscopic organism and they lack nuclei and other organized cell structure. So it's only one compartment. And bacteria is the plural form of bacterium. So while several bacterial species are pathogenic, which means they are capable of causing diseases, most are non-infectious and have critical role in decay, fermentation, nutrient recycling, nitrogen fixation, and many more. So bacteria are not always bad. So they are usually classified as gram-positive or gram-negative based on a microbiological staining procedure called the gram stain. So this is a very very rough structure of bacteria. So just to give an idea, and I know I'm sure everyone is aware of that, but still I wanted to put it because I want to touch all the basics. So these are the few organelles which are not membrane bound. So it's only one compartment, and that's the bacterial cell. These are unicellular, and this is the flagella, which are locomotive organelle for bacteria. And they are found everywhere under the earth to a distance of 400 meters, in the air at high altitude, at high temperature, around the craters, in the polar regions, within animals also. So they are like everywhere. And talking about animals, if we look at ourselves, so the bacteria are present everywhere in our body. So all over our skin, they are present in oral cavity, stomach, colon, vagina, our genital organs. So there are a lot of bacteria present. So this is just a time-lapse movie of just a growth of in an incubator after making a handprint on a medium where bacteria can grow. So here each dot represents some colonies, and we can see that contains millions of individual bacteria which are originated from invisible cell present on the hand. So whatever we can see in this movie on this black plate, these are all, they are always in our hand. So that is our the bacteria living on our skin in our hand. So how human we are. So as we are getting to know how much bacteria are residing within and on us, so that question comes to our mind, how human we are. So in terms of cell number, if we consider, so the bacteria and other microbial cells which are present in our body, so we can count that they are almost 10 times more than actual human cells of our body. So in terms of number, there are like about 39 trillion microbial cells residing in our body, whereas human cells are about 30 trillion. So if we see this picture over here, the red portion, which is that this is the total number of cells found in human body, and the red portion coming from us, like human, which is the 43% of the total cell count. So there are more bacteria. And on the other hand, if we look in terms of genes, so we humans, we have 20,000 human genes. And if we consider the genes coming from the microbial organism, so they are like, oh, I cannot count the zeros over here. So this is the pictorial representation. So this is the fraction of human genes when we count the total number of bacterial and also other microbial genes. So we are we can say that we are more microbe than humans. And these microbes residing in our body, they live there, and they play a very important role in our body. They are not always bad to us. So they protect against pathogens, pathogenic microbes. They help in human immune system development. It's a the code say the code presented here says that the microbiota plays a fundamental role on the induction, training, and function of the host immune system. In return, the immune system has largely evolved as a means to maintain the symbiotic relationship of the host with these highly diverse and evolving microbes. So they are continuously evolving, and they are helping us to shape our immune system. And apart from that, they help in digestion, supplies nutrients and energy, metabolize xenobiotics, induces development, synthesizes some vitamins, contribute to species evolution, and all sorts of things. So this is a very interesting field of research these days. And our faculty, Dr. Rachana Banerjee, will be talking about this very interesting topic tomorrow. So whoever is attending today's session, please don't miss tomorrow's session. So this human microbiome is very interesting, and these tiny microbes, they have so much effect on our body. So it's very interesting to explore. So come join Rachana Banerjee's talk tomorrow about human microbiome. So those are all the beneficial points from bacteria, but how can we forget? There are pathogen, notorious bacteria making trouble for us every day. So these are the few diseases that are caused by bacteria, like bacterial meningitis, pneumonia, skin infection, gastritis, food poisoning, eye infection, urinary tract infection, and so on. So there are pathogens who are creating trouble for us, and we need to deal with them. We have to. So there are good and bad bacteria both residing in our body. And the trickiest question is, in microbiology research these days, we have to deal with the bad bacteria only, keeping the good intact. So that will be our goal in microbiological research. Be it good or bad, all the bacteria, maybe they are residing in our body or in the environment, they have to struggle a lot. So the bad bacteria make us struggle a lot when they are infecting our body, but the bacteria itself, they have to struggle a lot. So they have to face many adverse conditions, nutrient starvation, high, low temperature variation, osmotic shock, and many more. But these tiny little organisms, they know how to deal with all these things. So they deal with them and happily live in our body, and depending on their nature, making trouble or doing good to us. So these things, these are achieved like surviving in adverse condition in ever-changing environment. To be able to do that, bacteria have developed a very sophisticated signal transduction mechanism by which they can actually sense the external stimuli. So that's called the reception. There are 3 stages of signal transduction. So they sense the external changes by receptor, which is called reception, and then they transduce that message to the internal other machineries, other protein machineries. So that process is called transduction, and according to the message, there are activation or deactivation of different cellular responses, and the bacteria deal with the change of environmental condition and stay happy in that new environmental condition. Yeah, so these are the 3 stages of bacterial signal transduction: reception, when a cell detects a signaling molecule from the outside of the cell, and a signal is detected when the chemical signal, also known as a ligand, bind to a receptor protein on the surface of the cell or inside the cell. So it could be inside the cell also. And then comes the transduction, when the signaling molecule binds the receptor and it changes the receptor protein in some way, and this change initiates the process of transduction. Signal transduction is usually a pathway of several steps. Each relay molecule in the signal transduction pathway changes the next molecule in the pathway. And finally comes the response. According to the change in the environmental condition, the signal triggers a specific cellular response to deal with the new environment. So to do that, for the signal transduction, bacteria have many sophisticated machinery. As the first step was the reception coming from the external environment. So most of those receptor proteins are situated in the membrane. So this is the membrane of a bacterial cell, and these are the cartoons of few well-known bacterial signaling proteins. So these are so that they have a membrane bound domain, and they have an extracellular domain which is involved in sensing and binding the external stimuli or the ligand, and they have an intracellular soluble domain to transduce the signal. So these are the three domains involved in the three processes of signal transduction. So the extracellular domain, that means which is protruding outward the bacterial cell membrane, and so that is sensing the environmental changes and binding to some ligands over here, and that is transduced through this the intramembrane part, and then comes the intracellular domain, which is involved in producing the final response. So these are the few major types of signaling protein membrane receptors that are involved in bacterial signal transduction. So we need to remember, so as multicellular organism like eukaryotic organism, so they need to deal with a lot, and their signaling mechanism are very sophisticated, but they have an advantage. They have compartments throughout their body. Like us, we have different kinds of cell in our body, and different kinds of tissues, and even inside a cell we have compartment which is facilitating the tight regulation of different cellular process. But for poor bacteria, they are just one compartment. So their signal transduction, their changes of intracellular processes need to be more intricately regulated. So they need, although they are a unicellular organism, but to survive in the hostile environment, maybe inside the host or outside in the environment, they need very tightly regulated system. So they need more tight control and more sophistication in their signaling mechanism. So these are the few major types of signaling receptor. So this is called the two component system. So I will be getting into detail of each type of each of these four types of signaling protein. So these are so I'm just naming them over here in this picture. So these are the two component system, and then comes the serine, threonine, and tyrosine kinases. So kinases play a very important role in cellular physiology, maybe it prokaryotic or eukaryotic. And then there another mechanism which is called second messenger signaling, where the signaling is done. So just the sensor make a small molecule inside a body in response to the external stimuli, and then that small molecule, which is known as second messenger, does the rest of the work. So that gives more efficient and tightly controlled regulation of different processes. And of course, there are methyl activating cellular proteins, which are regulating the flagellar motility, which is the a very basic form of um, response of bacteria in the change of environmental condition. So we have all known about, we have studied them in our school level biology, the flagellar motility. So that's the basic response, the chemotaxis, which are uh, which are regulated by these proteins, and that's the basic level of effect that bacteria shows in the change in environmental condition. And so in a study done, so they have counted the number of signaling proteins found in the bacteria, whatever bacterial genomes have to fully sequence. So they have counted each type of the signaling molecules, and they found that the two-component system, which consists of histidine kinases, depicted here as hiskin, and these are the topmost signaling molecule present in bacterial genome. So they outnumber every other signaling molecule. These are the primary signaling way that bacteria deal with changes in environment. And then comes the MCP, and then the second messenger signaling, and then the serine, threonine, and tyrosine kinases. So from this plot, we can see that the two-component system, where one component is the histidine kinases, they actually outnumber, and they. So we will begin our discussion. We will begin with the flagellar motility. And so methyl accepting chemotaxis protein, or in short MCPs, are the most common receptor responsible for bacterial movement, motility, translocation. So they sense the external changes of the stimuli, and they transmit that signal to a flagellar motor through a complex cascade of interacting proteins. So these arrows depicted here, they actually represent different steps. So we can see the activation of MCP, the signal is transduced to the flagellar motor. And not only that, MCP are also able to control the developmental gene expression. So they can control and regulate the different gene expression required for this motility. So, and this process is called chemotaxis, which is a directed motility by means of which microbes sense chemical cues and relocate towards more favorable environment. So, and this is important for bacteria. They basically do that for finding food, or move away from any harmful or poisonous substances. So the process are called the positive chemotaxis, which occurs if the movement is towards a higher concentration in the chemical gradient, and conversely, there could be a negative chemotaxis, where the movement will be in the opposite direction. If something harmful or poisonous bacteria sense that, then it will move away. So that harmful thing will be termed as chemorepellent, and where the helpful thing are termed as chemoattractant, which actually attracts the bacteria and pull it towards its higher concentration. And the flagellar motility, the motility are basically found two types of motility. These are all very highly regulated and multi-step processes, and depending on different steps at single transduction pathway, the rotation of flagellar could be of two types. It could be of counterclockwise rotation, so where the flagella is aligned into a single rotating bundle and causing the bacteria to swim in a straight line, and Apart from that, there are tumbling, that means which comes via clockwise rotation. So let the flagella bundle apart such that each flagellum points in a different direction, causing the bacteria to tumble in a place. So the direction of rotation are given for an observer outside the cell, looking down the flagella towards the cell. So that's the clockwise, anti-clockwise rotation are given in for an observer outside the cell, looking down the flagella towards the cell. So this is an example of, so far I have been talking about different steps. So this is a basic talk, so I'm not going into detail of each steps of signal transduction from MCP to flagellar motor, but there is a brief, this cartoon is showing very briefly how the action of different proteins. So the MCP here, it is sensing the external signal, it is binding to that. According to that, there is binding of a methyl group, which after binding that methyl group, there is an activation of different proteins, and the cascade of activation finally moves towards the flagellar motor and regulate the movement of flagellar, leading to movement in a bacterial cell. So the bacterial movements are important for bacteria to find food by swimming towards highest concentration of food molecule and to flee from poisons. But as the two types of movement of flagella show, that bacteria cannot move in a single direction. So they cannot move a straight line for more than 1 or 2 seconds. So after moving, so then due to the second type of signal, they change the direction and tumble, and finally again they assess them that they are moving towards a favorable condition or not. And then again, if they find that they are moving towards right direction, which is favorable for their existence, then they continue to move that direction. They change the direction by tumbling again, and then move to the in a straight line. So my words may sound confusing, so that's why I am putting over here a movie which is showing the movement of Campylobacter jejuni. And so this cell displaying flagellar movement here. So see, we can see that how the bacteria is continuously changing its direction, moving in a straight line for a very small fraction of time, then again tumbling, and then, so it's kind of erratic direction changes. And in this case, the chemotactic gradient is heading towards left of the screen. So therefore, so whichever, how many change of direction is happening, finally they are moving towards the direction which is favorable for their existence. And this ability of Campylobacter jejuni is the main cause of causing diseases. As we know, that Campylobacter jejuni is one of the major causes of food poisoning worldwide, and depending on its motility, so they are, it depends, motility is the major factor of Campylobacter virulence. If the flagella is not there, they cannot survive in our body and create the diseases. So these kind of signal transduction are very much required for this pathogen to cause disease in our body. So we have seen that bacteria is unable to choose the direction in which they swim, and they are unable to swim in a straight line for more than a few seconds. And actually the chemotaxis that directs the overall motion based on the chemical gradient. And bacteria actually use temporal sensing to decide whether their situation is improving or not. So it's really hard to imagine for a tiny little organism like bacteria. Within a few seconds, they are measuring the concentration outside, and they are assessing it and deciding that it is in their favor or not, and depending on that, they are changing their direction of movement. So these kind of chemotactic responses, such as like forgetting the previous direction and choosing a newer movement, resembles actually the decision making abilities of higher life forms with brains that process sensory data. So so this is we can see how like how the multicellular organism advanced level cognitive skills are coming. So this is the uh, primitive signal transduction and decision making ability we have seen in bacterial cell, which is seen in a more complicated way in the higher forms of life. So I find this really fascinating. So moving on to the next one. So we learned a little about these methyl accepting proteins. Then we'll move to the major resources of bacterial signal transduction, that is the two-component system. So what are two-component systems? Two-component regulatory systems serve as a basic stimulus response coupling mechanism to allow organism to sense and respond to changes in many different environmental conditions. So as the name suggests, it consists of two different components. So there typically the first component is a membrane-bound histidine kinase. So kinases are the protein which undergo phosphorylation changes, which means a phosphate groups coming from a high energy compound like ATP, a phosphate group is coming and binding to some amino acid residues found in the protein, and in this case this is the histidine. So histidine kinases get phosphorylated by the one of the high energy phosphate group in ATP, and the phosphate group binds to the histidine residue found in the protein. So that's how the name comes, like histidine kinase. So the first component of the two component system is called the membrane bound histidine kinase, that actually senses a specific environmental stimulus and a corresponding response rate. And there is, and the second component, obviously, there comes, that is called a response regulator. So the stimulus that is sensed by the histidine kinase, it passes down that signal to another protein called the response regulator, which is actually doing the changes in the cellular response, which is mediating the signal. The function of the histidine kinase is to sense the signal and pass it on to the response regulator, and it takes the response regulator takes the responsibility to work accordingly the signal. So it changes mostly all the response regulator bind to the DNA, and they brings changes in gene expression depending on the signal that is sensed by the histidine kinases. So these two component signaling systems are widely occurring in prokaryotes, whereas only a few two component systems have been identified in eukaryotic organisms. These are mostly found in prokaryotes, but there are some variation. So, and talking about the histidine kinase, it is basically it's a modular stimulus response system. So the histidine kinase has two domains: the conserved kinase core and a specific sensory domain. The sensory domain, which actually sends the input signal, and this signal is transmitted to the transmitter domain, and histidine kinases, they transfer that signal, which is in form of a phosphate molecule, to the receiver domain of the response regulator, which also have two different domains, the conserved receiver domain and the specific effector domain, which will which will act according to the signal, and which is mainly binding to the DNA sequence and regulating some gene expression which are required for the newly acquired environmental condition. So histidine kinases are multifunctional, typically transmembrane protein of the transferase class, and they play a role in signal transduction across the cellular membrane. So as I mentioned, the signal transduction occurs through the transfer of a phosphoryl group from a ATP or adenosine triphosphate to a specific histidine residue in the histidine kinase, and that is called an autophosphorylation, where the protein itself is taking off a phosphate moiety from a high energy phosphate molecule, which is ATP here, and the phosphate is binding to the kinase itself, and that reaction is called autophosphorylation. Subsequently, the histidine kinase catalyzes the transfer of the phosphate group on the phosphorylated histidine residue to an aspartic acid residue on the response regulator. So then comes the transfer of the phosphate group and which to the response regulator, and the acceptor moiety in the response regulator is an aspartic acid residue. So basically a phosphate group binds to the histidine residue in the kinase, and this phosphate group is getting transferred to another protein which is called response regulator, to an aspartic acid residue of that response regulator. So phosphorylation causes the response regulator conformation change and usually activating it to attach the output domain, which leads to the stimulation of expression of target gene. So upon phosphorylation, the conformational changes that happen to that in that response regulator protein, and that is making it able to bind the specific DNA sequence and regulating the expression of the target gene. So the level of phosphorylation of the response regulator controls its activity. Some histidines are bifunctional. That means they catalyze both the phosphorylation and dephosphorylation of their cognate response regulator. You mean when the phosphate group is transferred to the response regulator, it get activated, right? But when the external signal is gone, the stimulus is gone, then it needs to turn off. And so that case, the phosphate group transferred to the response regulator needs to take off, and that is done by a phosphatase activity. So kinase is addition of phosphate, whereas the phosphatase activity is removal of phosphate group from a protein. So in some cases, the histidine kinases are capable of doing both that functions, which is called bifunctional histidine kinases. But usually in case of two component system, the phosphorylation reaction is labile. So the phosphate group attached to the response regulator does not stay for longer time. So the need of phosphatase, the need of dephosphorylation by some active phosphatase is not there for most of the cases. So as if we remember the plot where we find the abundance of these signaling domains, signaling molecules in throughout bacterial genomes, we found that the histidine kinases were mostly abundant. And here just a brief account of genomic distribution of these histidine kinases in different bacterial species like E. coli. We can count about like 30 to 32, and In Mycoplasma, there are none. In Bacillus, there are 70, and Helicobacter pylori, a very well-known notorious pathogen, have only 11. In this context, I would like to mention one thing. We know about IQ, like intelligent quotient, for all our human being. Even bacteria have IQ. So the term bacterial IQ is there, and it is measured by the number of signaling molecules or signaling proteins present in a bacteria in the whole genome. So the more the number of signaling molecule, the bacteria will be called smarter, and the less the number, they are dumb. So, and it has been observed that the bacteria, the free living bacteria, those bacteria live in the environment, and they have more number of signaling proteins, moieties, and so they are smarter than the pathogen or parasite living inside somebody. So we can see in terms of only histidine kinase, the free living bacillus have more number of kinases than the pathogen. This is a very small instant, so the conclusion is obviously not drawn with this few number of, so it has been widely studied, and it has found that the free living bacteria have more signaling proteins, modules than the parasitic or pathogens. So pathogens are actually dumb, who residing in our body and doing harm to us, whereas the free living bacteria are smarter. So one exception in this case is E. coli. So if we can see the number of signaling that here only we can see the histidine kinases, but in total the number of signaling moieties are less in E. coli. It's an exception, and so maybe that's why E. coli is like opportunistic. So it can stay outside in the environment, in our gut. So it can be commensal, it can be pathogen. So it can change its lifestyle according to the environment. So this is so maybe it might look dumb, but in function it might be smart. So that's an exceptional case. Otherwise, all the pathogens, the notorious bacteria, have low IQ, and so maybe that's why we can overcome them. And the free living bacteria, which is good for us, and they are having the higher IQ. Coming back to histidine kinases. So if we look at the structural details of these histidine kinases, they most are periplasmic, most have periplasmic membrane receptor. They function as homodimers. The autophosphorylation reaction is a bimolecular event. So usually they have a periplasmic N-terminal binding domain, and then a transmembrane domain, and then a linker domain, and a histidine containing phospho-transfer domain, the C-terminal kinase core. So these are the few domains present in the histidine kinase, and when they are active, they come together, they form a homodimer, and they get activated by the process of autophosphorylation, which is addition of the phosphate moiety to the histidine residue. Some histidine kinases, there are exceptions. So some histidine kinases are soluble. They are not membrane bound. They don't have this transmembrane domain over here. So the sensing domain, which is the N-terminal domain that senses the external stimuli, usually a periplasmic receptor. This is the periplasmic space over here, and the portion of histidine kinase, this is the N-terminal sensing domain. So not always, as I mentioned, in many cases, the ligand or stimulus is unknown, and so in many cases, this external N-terminal domain is absent in the histidine kinases. So, and then the transmembrane and linker domain. So this is critical for propagation of the signal, which is coming from this portion of the protein and from the periplasmic binding protein to the kinase core, which is this, where the active site, That means the histidine residue is present over here. And there are the kinase catalytic code, which is about 350 amino acid in length. So they are actually the dimerization domain. So the activation and the dimer formation, all this is happening at this domain, and they are the ATP binding and the phospho-transfer domain. So the phosphatase activity, if there is any, that is also found in this domain. And then of course the histidine containing phospho-transfer domain, which is around 120 amino acid in length, and the histidine residue is present there, and no kinase or phosphatase activity is found in this residue. So the histidine residue present in this domain get phosphorylated, but the kinase the kinase activity, that means which is coming from binding the ATP and transferring the phosphate, is not found in this domain. So depending on the presence of the extracellular cellular domain, the histidine kinases are divided in two classes. Class 1, which have this extracellular periplasmic signal accepting domain, but some histidine kinases do not have that domain. In that, those are named as class 2. In that case, the membrane bound domains actually work as the sensor domain. They sense the external signal and pass it on to the downstream cytoplasmic domain. And looking towards the response regulator, they have the N-terminal receiver or regulatory domain, which have the aspartate residue, which will accept the phosphate donated by the histidine residue present in the kinase. And then they have the C-terminal effector domain, so which is actual DNA binding transcriptional regulator domain. And they might be involved in other than DNA binding in protein-protein interaction, or in some cases enzymatic activity. And so catalyze the transfer of phosphoryl group from phosphohistidine kinase to conserved aspartic acid. So they brings the conformational change in the response regulator, and then the response regulator is able to bind to the actual active site. So these are the few types of two-component system where there is involved in different kinds of regulation. This is involved in osmoregulation, the typical histidine kinase, which have all three domains, and the response regulator. But the transfer is happening once, but the phospho-transfer reaction can be a relay transfer, which is called the phosphorylase system. So here the histidine residue over here is binding to the phosphate, and then this phosphate is transferred to another aspartate residue present in the kinase only, and then again from there the phosphate is traveled to the histidine residue in that particular kinase, and then the final transfer to the response regulator is happening. So this is called the phospho-relay system. So adding this kind of complexity provide for multiple regulatory checkpoints. So these, as I mentioned, they are the single compartment system. So this kind of complexity, they actually provide the multiple regulatory checkpoint. So here there's only one checkpoint, but here there are many steps. So another kinds, they are involved in. So there are two kinds of histidine kinase. They are coming together and acting together on a single response regulator, and then finally through phosphorylase system, they are actually transferring the phosphate residue to the actual response regulator. So they are working hand in hand. So both of them has to be active. They has to be involved in this reaction, and so that is also adding an certain level of complexity to the situation. And so here, so we have seen the example of two different histidine kinase phosphorylating one single type of response regulator. The reverse could also happen, where one kind of kinase can phosphorylate two different kind of response regulator. Yeah, this is the phospho-transfer. So the whole point of all this complexity is to regulate the signal transduction. So the signal pathway provides steps at which the flow of information can be modulated. The regulation of histidine kinase is coming from the autokinase activity, either stimulated or repressed by specific stimulus. And the response regulator phosphatase activity of the kinase that can be also modulated. And the regulation of response regulator activity can be done by controlling the phosphorylation by the cognate histidine kinase or dephosphorylation by some specific phosphatase. So these are the steps where further regulative pathway could be involved, regulatory pathway, and there are making it more complex and adding some extra checkpoints. So, and the stimulation of intrinsic autophosphatase activity, which is also present in this response regulator. Stimulation of that activity, which will render the response regulator inactive, so that can also be controlled. And obviously inhibition of the phospho-transfer from the kinase to the regulator, and the regulation of the expression of two component proteins. So the expression of these two components can also be regulated by some other proteins. So these are some external regulatory mechanism that can be involved in regulating these process, which is performed by this type of two component systems. So this is an example of a two component system found in, which is known as 4PQ, where we can see the 4Q is the membrane bound kinase, and the signal, external signal here is the concentration of divalent magnesium ion, and the response regulator here is 4P. So by sensing the low magnesium ion concentration externally, 4Q is phosphorylating the 4P protein, which in turn regulates the expression of different genes involved in different process. Basically the magnesium transport, which is MGTA, MGTCB, and this. And other than that, they are involved in gene expression of LPS modification. They actually gives the resistance to antimicrobial peptides and transcriptional regulation of their own expression. That is also done by the response regulator itself. And so to further add the level of complexity, there could be a crosstalk between two types of two component system. Here we can see that 4P is regulating expression of another two component system, which is PMRA, PMRB, which is involved in sensing the concentration of iron externally and regulates many proteins involved in the bacterial survival, virulence, and other genes transcriptional regulation. So there is a crosstalk between two different two component system. The one response regulator from this first 4PQ two component system is regulating the function of another two component system. So this kind of they are involving with each other and enhancing the complexity and more giving more tight regulation to cellular reactions of bacteria. So the third type of signaling moiety we will be talking about are serine, threonine, and tyrosine kinases. As the name suggests, transfer of phosphate group is involved here also. So the serine, threonine, and tyrosine kinases transfer a phosphate group from ATP or GTP onto a specific serine, threonine, and or tyrosine residue of a protein structure. So the phosphate group found in the ATP, so this is coming and binding to this protein serine, threonine or tyrosine, which have the free OH group over here, and the phosphate is binding over that. So that reaction is called the kinase activity. And since the phosphorylating moiety here is either serine, threonine or tyrosine, these kinases are called serine, threonine or tyrosine kinases. So typically phosphorylation functionally activates the substrate to perform either a specific activity or cellular localization and or transfer the phosphate group to a downstream effector, initiating a cascade of signal transduction reaction. So in eukaryotic system, we are very much familiar with this kind of cascade of signal transduction system where one kind of kinase are activating other kinase, and then it is going so on and on. But that kind of reaction could possible in bacterial system, but in a few numbers. So the reverse reaction of dephosphorylation actually restores the activator and effectors to their initial functional state. So for this ST kinase, the kinase and phosphatase reaction, the phosphorylation and dephosphorylation reaction occurs hand in hand. So the kinase, there is a dedicated phosphatase which actually cleaves that phosphate group, which is added to the kinase and uh, turns it off or on. So the kinase and phosphatases function as an on-off switch, modulating specific signal transduction pathway. So it can be either way. The addition of phosphate may render the protein in an on state or an off state, and reversibly, phosphatase activity could put it in an off state or on state. And so bacterial serine, threonine, and tyrosine kinases, they dynamically coordinate several cellular processes. So they act as cell cycle checkpoint regulation. So in eukaryotic system, the cell cycle checkpoint process are regulated very well, and they are also coordinated with cell growth. The existence of highly intricate signaling networks ensured by quality control and checkpoint proteins that coordinate this sequential event. They trigger cell cycle arrest when the things go wrong. So that is when the cell cycle need to be arrested. And as I mentioned, in bacteria, the different processes are more interwoven. So consequently, the accurate execution of each step relative to other in time and space is crucial. And so it involves a strict regulatory mechanism, and this serine, threonine kinase and phosphatase action provides that strict regulation. These are also these kind of kinases are also involved in global gene expression and cell wall metabolism. So these kinases actually sense cell wall related processes and subsequently mediate the regulation of cell wall biosynthesis, and they are involved in modification of transcription factor and other known transcriptional regulatory mechanism. And sometimes they are involved in phosphorylate elements of two-component system. So this is an example of serine threonine kinase found in one of the notorious pathogen called Mycobacterium tuberculosis, which is the causative agent of tuberculosis. And the sensor domain, which is extracellular sensor domain found in these kind of kinases, are called pasta domain. Not the edible one, but the full form of this pasta domain is peptidoglycan and serine threonine kinase associated. So that's the full form pasta, and these domains are involved in peptidoglycan, which is the major constituent of the cell wall and membrane of bacterial cell. So here there are two serine threonine kinase from Mycobacterium tuberculosis. One is PK and B, which have these pasta domains. They are pasta repeats organized linearly, and whereas this kinase, which is called as PKNG, they have a highly symmetric six-bladed propeller formed by the extracellular domain of this PKNG kinase, and this these are the regulatory domain to represent the intracellular catalytic kinase domain. So they actually sense the external signal, and the intracellular kinase domain act according to the signal. So this is a reaction done by the PKNB. So it is getting activated by the stimulus, and autophosphorylation undergoes autophosphorylation, and these phosphorylated PKNB then become activated and phosphorylated another substrate, or it can also phosphorylate a phosphatase. In this case, it is PSTP. This is just a case example of Mycobacterium tuberculosis, where this serine threonine kinase PKNB phosphorylate the phosphatase PSTP, and actually this phosphorylation activates the phosphatase, and then which actually in turn dephosphorylate that kinase over here and making it inactive. So this is a very timely regulated system where this activation stays for a very limited period, and the cellular processes accordingly are activated. So this is the activation model of a PKNB from Mycobacterium tuberculosis. So in the presence of ligand, two or more PKNB monomers bind to a single ligand molecule, and through their extracellular domains, and this brings two extracellular domains close together, and they actually form a dimer, and that activates this protein, and it consequently it is taking up the phosphate moiety and getting autophosphorylated. The activated kinase can directly phosphorylate a downstream protein target like this. So this, after activation, it get the dimer is removed, and this activated protein can phosphorylate another protein which is involved in regulating the cellular process, or it can also target and activate another soluble kinase through the formation of asymmetric and front to front dimer, and which will then phosphorylate downstream targets as part of the signaling pathway. So all sorts of multi-step regulation happening over here also. So the bacterial serine threonase kinase, so the all and the two component system, we can see the basic steps are phosphorylation and phospho-transfer. But there are few differences. So the basic steps are similar, although there are few differences. One is obviously it's a two component system, and here everything is done by the kinase itself. So it is actually, it can sense the signal and can generate the cellular response, but there are a few differences, also few more differences. So all these bacterial serine threonine kinase, they so there are few bacterial serine threonine kinase which have high similarity with eukaryotic kinases, whereas two component system are mostly found in prokaryote, and so homologs of this prokaryotic two component system are not found in eukaryotic system. So that's the major difference, and depending on that, the bacterial serine threonine kinases are they are separated in two classes: eukaryotic like STK or which are different in sequence from eukaryotic kinases. So the eukaryotic like STK or the ESTK are the evolutionary predecessors of eukaryotic serine threonine kinase. So the nature actually selected this kinase, and they have selection advantage. They can efficiently transfer the signal, extracellular signal, and regulate the cellular mechanism. So they are selected in the evolutionary procedure and found in the eukaryotic cells also. And another difference that we can find in between two component system and serine threonine kinases, the phosphorylation is very stable in serine threonine kinases. So they require an active dephosphorylation to facilitate the reversible regulation, whereas the two component system, phosphorylation is mostly and relatively labile, and a specific phosphatase is not necessary in most of the cases to remove it. So other than these few differences, the basic thing in these two serine threonine kinases and two component system in these two regulatory molecules is the phosphorylation reaction. And coming to the role of the serine threonine kinase towards disease development, or we can say virulence. So, these STKs regulate the peptidoglycan synthesis, and that can be disrupted by some inhibitor, which will render the pathogenic bacteria sensitive to those agents. So if we inactivate these, so these are very popular drug targets. So targeting these STKs can render many pathogenic organisms. So we can get rid of them if we target these serine threonine kinases. And but some of these serine threonine kinases are essential and thus have been the subject to intensive study of antibiotic targets such as PKNB from tuberculosis. And even in bacteria containing non-essential homologs, deletion mutants leads to severe distortion in cell shape, and these changes, which may permit viability in normal laboratory condition, but they could result in a disadvantage in the environment. So and rendered the bacteria unfit for survival. So serine threonine kinases play such an important role in most of the bacterial physiology. So targeting them by some drug, inactivating them, we can actually get rid of that bacteria. And so talking so much about phosphorylation, dephosphorylation, I should mention here a very important field of biology these days. We can see how important the phosphorylation, the reaction involving phosphorylation are to bacterial physiology and also eukaryotic physiology. So there are two terms that have evolved these days, which is called phosphoproteomics and interomics. Sorry, so, excuse me, interactomies. Mix. So what is phosphoproteomics? It is the large-scale profiling of phosphoproteins and phosphosides by mass spectrometry-based proteomics, and these are very helpful to identify kinase-specific substrate and understand the functional dynamics of phosphorylation network. So most of the signaling networks are involved with this phosphorylation reaction. So measuring the phosphoproteins, by marking the phosphosides and identify their targets. So that is a very important field of research these days to understand both the prokaryotic and eukaryotic physiology. So phosphoproteomics have evolved as a very important field of research. And so is interactomics. Interactomics actually identifies the kinase protein interaction, revealing not only the potential substrate, but also anchor, modulator. So all these processes are very much interconnected. And ever-growing data from large-scale phosphoproteome studies in bacteria coupled with identification of kinase, phosphatase, substrate, and modulators. They provide actually important insight into cellular regulation. So the powerful, so these two fields, phosphoproteomics and interactomics, are powerful and complementary to each other. And a study of these two fields can provide a blueprint of kinase and phosphatase-centered signaling network, which is mostly it is in case of higher organism, and gives an important insight into cellular regulation of protein activities, their interaction with other proteins, and their localization in cell. So coming back to our topic, we have one left in the signal transduction machinery, which is involving the second messenger signaling. So this is a brief account of the second messenger pathway over here. So the external stimuli, whatever it is called the first messenger in this process. It could be the light, change of temperature, the ion concentration, whatever. So that some protein will sense that changes the first messenger, and in response to that, some other small molecules are created in the cell, which are called second messenger. Then this small molecule, this second messenger, actually bind to the effector proteins, which could be DNA, RNA, or protein complexes, and then the regulation of different cellular processes are occurred. So from all previously discussed processes, so there is a huge change in the mechanism. There is a new molecule is being synthesized in response to change in external stimuli concentration. So these are the few bacterial small molecules which acts as a second messenger and synthesized in response to the change in environmental signal. So like NAD, NADP, so this is a timeline. There is cyclic di-GMP, which have immense role. There is cyclic di-AMP. These are all nucleotide-based second messenger. And if we look at the functional, so we can see that these all three tiny small molecules are capable of doing many cellular function. They are regulating many cellular functions. Like CMP, it is regulating the virulence, biofilm formation, and cyclic di-GMP is a very much important second messenger. It actually regulates sessility, motility, virulence, biofilm formation. And there is PPGPP, which actually regulates DNA replication, virulence, transcription, translation. And there is cyclic di-AMP, which regulates antibiotic resistance, motility, virulence, pyrrolation, cell wall biosynthesis, growth, etc. Many cellular process we can name. So we can see that this mechanism, like the second messenger signaling, is very popular in bacterial world, and these tiny small molecules, they are regulating many important bacterial cellular processes. So to get into insight of some of these, so I'm choosing this cyclic di-AMP, which is comparatively new in this field. It is identified in 2008, and this is the structure of cyclic di-AMP, where two molecules of AMP are cyclized. So they are forming this phosphate bond here and making this structure. And it was first identified from Listeria monocytogenes, which is a pathogen to us, and it is actually identified as a secretory molecules. It is actually secretes out from the bacteria when this bacteria is inside the host cell. So this is the inside the host cell cytosol, the blue one, and that's the nucleus of the host cell. So when the bacteria, the L monocytogenes is inside the bacteria, it makes this small molecule, it sends some external signal, makes this small molecule and secrete it out, which is sensed by the human host, us. So they sense it as the presence of that particular bacteria and generate immune response. So this is an immune activator. This is actually acting as a immune activator and generating the immune response. So this is how these second messenger are helping the bacteria to cope up in the host environment, but we are being smarter than them. We actually use those particular molecules and as a marker of presence of those bacteria and generate the mechanism to kill them. The immune system is known for that, right? So that's one part of the story. So cyclidine, apart from doing that, cyclidine actually regulate many important cellular physiological process. So they are involved cyclidine homeostasis. So I should mention here the concentration of these small molecules, the second messenger, are very tightly regulated. Both low and high concentration are bad for bacteria. So like cyclidine homeostasis, so there is an optimum amount of second messenger concentration should present, and that is the homeostasis, and this is required for actual bacterial growth. So if you lower down the cyclidine level, we can see the growth of, this is a bacterial growth curve. So this is the wild type which have the optimum amount of cyclidine, and this is one which have lower amount of cyclidine, and it is, we can see it is growing less. It is actually controlling cell size, cell wall stability. So we can see here, so this is y-axis here, we are measuring amount of lysis. When the cell ruptures, it is called lysis. So if the stability of the cell wall is lower, the cell tends to rupture. And we can see in a cell where the amount of cyclidine is low than normal, they tends to lyse, this tends to rupture, which is saying that cyclidine right amount is required for cell wall stability, maintaining the cell size. It is involved in responses to different stress, which is obvious because it's a second messenger. Its function is to sense the external stress and act accordingly. So in absence of that, the bacteria are dying more. So this is less bacteria with less amount of cyclidine, and we can, and this is y-axis, this is the amount of that which is measured by zone of inhibition. So higher the value, that means more bacteria are dying. So if you lower the cyclidine, we can see in terms of stress application, the bacteria is dying. And last but not the least, this is very much required for establishment of successful bacterial infection, as this cyclidine is found in many important bacterial pathogens. So we have seen that if we deplete that pathogen from the normal cyclidine level, or if we even increase the normal the level of cyclidine than normal, the survival of those bacteria, those pathogen becomes lower. So this is a measure of survival of bacteria inside a mouse model. So the mouse were infected with this bacteria, and the wild type with the ideal amount of cyclidine are surviving very well, where there are almost 4 logs of difference in number of bacteria surviving when the cyclidine level is low. So as I mentioned, the cellular concentration of cyclidine is very tightly regulated, just like other second messenger small molecules. So there is a synthase, which is here in this case is diadenylate cyclase, which is known to form the cyclidine by from 2 molecules of ATP and a degradase, which is the phosphatase, which actually cleaves the cyclidine and generate this linear PAPA molecule. So and as specific for cyclic DMP, there is a transporter which is involved in efflux. That means taking that that signaling molecule out of the cell. So this is a transporter which is taking this cyclic DMP out of the cell. So that is also regulating the intracellular concentration of cyclic DMP. So this one is making the cyclic DMP, this is degrading it, and this is taking it out. And controlled regulation of all these proteins I actually regulating the cyclic DMP levels. And all these proteins have extracellular domain, membrane bound domain, which are sensing the external signal, and according to that they are regulating the actual intracellular concentration of this molecule, which then binds to different affected molecules, may it be DNA, RNA, or proteins, and regulate the cellular physiology. So this is a, if you find it complex, I would very quickly walk through this picture over here. So this is few of the this is a cartoon of few of the proteins that are involved in cyclic DMP signaling. So here is the synthase, which is synthesizing the second messenger. See, this is one another type of synthase. This synthase is involved in maintaining the DNA integrity, and here is the, so there cyclic DMP is involved in potassium transport. So here is the potassium transporter which is binding to cyclic DMP. Then another potassium transport. Cyclic DMP is involved in fatty acid metabolism. This is binding the DARR protein. This is there is another protein called PSTA, which is known to involved in nitrogen metabolism, and this is a ribose switch is involved in regulation of differential gene expression. Cyclic DMP actually binds to that also and regulates its activity. And above that, cyclic DMP actually binds to a very important central metabolic enzyme called pyruvate carboxylase. So this is, just to remind you, this is an enzyme involved in the TCS, so the basic carbon metabolism pathway, and that is a very vital enzyme in that process, and cyclic DMP actually binds to it. And there are phosphatases which are cleaving cycle and maintaining the intracellular level. So talking about the important metabolic regulator pyruvate carboxylase, let's this is a very interesting movie which shows, this is a picture 3D structure of pyruvate carboxylase crystal, and here the U-shaped molecule is cyclic DMP. So we can see here when cyclic DMP binds, so this is the unbound state, we can see the molecule is moving freely. See, so this is the unbound pyruvate carboxylase, the molecule is moving very freely, but as soon I believe I got disconnected over here.
Your voices were trailing, so yeah. Yes, you can start from here.
Okay, okay.
Yeah,
Yeah,
please
so
repeat from your U-shaped molecule. Yeah.
okay.
Mhm.
So yeah, so this is crystal 3D structure of pyruvate carboxylase crystal, and here this is the unbound protein. We can see it is moving freely, and as soon as cyclic DMP comes and binds, this is a U-shaped molecule over here, the enzyme is locked, the movement is freezed, and that freezing actually makes it uh, the activity is inhibited. So cyclic DMP is actually inhibiting this pyruvate carboxylase activity by locking it in a particular conformation and freezing it, and the activity of the enzyme, the metabolic activity is inhibited. And another very interesting point I would like to mention as the so how integrate this signaling reaction could be. So we have seen that the both like high and low level of cyclid-IMP was bad for this enzyme activity, because this kind of regulation of pyruvate carboxylase by cyclid-IMP was very much important for that bacterial cell survival. And we have seen that if you go on decrease the cyclid-IMP level or increase it, the survival. So this is the y-axis over here, which is depicting the survival of the bacteria. So if you go on increasing, the survival increases at certain point, and after that it loses. So we can clearly see how the intracellular level of this particular second messenger is important. If you deplete from the ideal level, the survival goes down, and if you increase, the survival also goes down. So I guess for the first session, we have talked a lot about bacterial signal transduction, and these are basically the building blocks of signal transduction. So this is the actual signal transduction processes are more complicated. They all do crosstalks with one another, and so and there is a like complex network present, but the building blocks are only these few things. And so the pathogen in terms of the disease causing, all these integrated the networks I talked about, and so including so this sense, they are involved in sensing the change in the environment, maybe it the free living bacteria or the pathogen, so where they sense the hostile host environment and act accordingly. The change alter their behavior in order to better survive. So pathogenesis can also be viewed as a bacteria simply adapting their behavior to survive and thrive in a new environmental niche, which is the host organism. And in order to survive, it has to, there are multiple processes, like it has to and so the route of entry, the mode of transmission, and the host susceptibility, these are the few factors that decide the survival of the bacteria in the host organism, which is called as host pathogen interaction. And several factors are required to survive in the hostile host environment and evade the host defense. Obviously the host has a defense mechanism, and damage the host tissue, creating diseases for us. So understanding all this signaling system and the interaction of pathogens with host is very much required, very important, because only in that way we can find a way to fight against these pathogens which are causing the infectious disease. So this is why we need to like study this field and to overcome this kind of pathogen. So I think I should stop here. And so thank you for your attention and being with me. So if you have any further question, you can contact. This is my email ID over here. And as I mentioned in previously during my talk, so there is an upcoming webinar by Dr. Rachana Banerjee about human microbiome and its influence on health and diseases. So whoever is attending, whoever is present here, I think it would be a great and interesting talk to attend. And not only that, we have another coming webinar by Dr. Sujay Chatra, computational genomics. So that is, I believe, a more interesting talk to watch. I hope we can find you there, and don't miss those talks. And thank you once again to everyone. And I think if there are any questions, we can answer them now. Dr. Ganguly, are you there?
Yes. Yeah. Thank you very much for such a wonderful talk. It's very insightful.
My
I'm
pleasure.
sure our participant enjoyed every tips and bits. So there are quite a few questions from an anonymous attendee. Ma'am, how does pathogen affects human body? Is is it that first a pathogen protein affects human protein, which in turn affects their neighborhood proteins through
So
interaction?
your voice is also breaking. Can you
Yeah,
repeat
yeah,
it? Yeah. I the network
yeah,
connection
yeah, yeah.
is
So,
making trouble.
yeah, can you can you look at the chat section? So I'm just reading this out. Okay. His or her question is, ma'am, how does pathogen affects human body?
Okay.
Is it that first a pathogen protein affects human protein, which in turn affects their neighborhood proteins, though interaction? I think through interaction. Okay.
Which in turn
That
affects their neighborhood protein.
yes,
Mhm.
that's how it gets transmitted.
Yeah. Uh, Pathogen affects human body. So actually, what the pathogen wants to survive inside the human body. So they don't want to make harm to us. Okay. So in terms of their survival inside our body, they are making different transitions. They are making different interactions over there. That is, they want to survive. In that course, we are all selfish, and pathogens are working like selfish. They produce some kind of some pathogen produce some pathogens because of their survival. Yes, they get into, they invade our body, and in terms of their survival, they affect our body. Yeah, it could be via so every cell processes are different proteins, that is the first kind of interaction. So yes, but the
Okay.
transmitted part, that is not true, because it's just a survival of the bacteria. So how the pathogen is getting into your body, that's the part of transmitted. So it is maybe interacting with some cellular resource getting into, then it's yes, then it is between protein interaction, because if it is not, then it is not. But mostly the major cellular processes are regulated by the protein, so it is yes.
Okay. Okay. Thank you. The next question is, ma'am, how gene mutates? Does it depend
Mhm.
on some external stimuli? If it is, then what is that?
Mutation is a random event. So basically, when the DNA replicates, if there is any faulty replication, although there are many checkpoints, mutation is a random event. It happens on its own. Some chemicals may stimulate them. Presence of some chemicals may stimulate where the replication machinery or other machinery which are maintaining the fidelity of the DNA is not working properly, then it could be induced by that. Otherwise, mutation is spontaneous. The gene are mutated spontaneously.
Okay, yeah, now I know how gene mutates. Okay, so if dear participant, if you have more question, please write it down in the question and answer section. Okay, our next question is also from an anonymous attendee. Does gene expression values can control gene mutation?
um, you mean like how if there are more expression, there could be more mutation? So
More
that's,
I
there is
think
no
so.
direct
Yeah.
correlation. Yeah, no such correlation can be drawn that if gene is highly expressed, there is higher chance of mutation. That is not true.
Okay. So let's see if we have any more questions. So again, participants, please feel free to write your questions. Dr. Shurika will be happy to attain the questions. Let me check. there is any other
Yeah, I
questions.
guess in any way, signaling thing is so complex that makes you feel sleepy. So I guess there are no more questions out there.
No, no, you explained this very, very efficiently. So I'm not from the biology background, but I understood that. Okay.
Oh,
So
so
then again we announce tomorrow we have another very interesting talk. Dr. Rachana Banerjee will speak on the human microbiomes, and the day after tomorrow, our head of the department, Professor Sujoy Chattopadhyay, will speak on the genomics. So please, if you are interested, do not miss those two talks. And thank you very much, everyone, for attending.
yeah, thank you. Yeah,
Thank
thank
you.
you from my side
So
also. Mhm.
yes, so I'm ending this meeting and
Mhm.
have a have a great evening. Bye.
Bye.